Okay, guys. Sorry for the delay. Start. So let me ask the back row. Can you guys hear me? Because my evaluator said the major problem is that they couldn't hear me in the back. I don't know if it's that they're old people who are evaluating me and they've been to too many rock shows, or what. But if you cannot hear me, you need to tell me, because there's a way to set up with a microphone. We can get sound effects, costumes.
Yes.
I had a friend who worked at Poly. I don't know who's from Baltimore. Baltimore Polytechnic. I don't know. Doesn't matter who school that's good. And she worked there, and she was awesome. And she actually had a contest for like who could make the best life-size DNA. And someone brought in DNA with made out of a weave wrapped around Barbie heads. Awesome. I'm just saying. There's so many ways we can know this, right? Histones, Barbie heads. Anything like that?
Yeah.
Anyways, she's no longer there, though. She moved on. Okay. Quick business. Exam 1, I finished grading it at 1 in the morning last night. I'm behind schedule on that because of Wednesday. Those of you guys might think, wow, but we weren't here on Wednesday. Why weren't you grading our exams? Well, it's because I had 4 kids under the age of 5 in my house. Not all of them mine. So most of them mine. One kept whining like Caillou, and it was a lot. No, actually, he's a sweetheart, but kind of. So I did not do anything. And I hurt my back, I think, from lifting heavy children. I don't know what I did. So I was up until 1 in the morning because I was like, I promised by Friday. I'm like, so they're done, but there is some differences between the essays between the 3 sections, and I want to make sure the averages are the same to be sure it's fair. So that's why you're not getting them back right now. Probably after recitation, I'll sit down and work that out, and I'll post the grades today at some point by end of day on Blackboard. The grades, without being adjusted at all, were fantastic. I would say I've never seen so many over hundreds, which is great. I've also seen there are some that are really scary. So those of you guys who would categorize yourself as having a scary exam, come see me, because there's a huge disconnect going on. And if it's just that, like, you're like, I don't care, I just need to get through this, then whatever. But if you're like, I really want to do well, come see me.
So the majority of us did well.
You did horribly. You failed. Shouldn't you get that if you ask me? No. Okay. Wednesday recitation. I know you're very sad that you had to miss recitation on Wednesday. We're going to make it up next Wednesday. You guys already read the papers. You've already looked at everything, right? So you have until next Wednesday to do that, and you're going to double up. I want you to have that quiz, so because I could just say forget it, but then you won't have enough quizzes to drop. So I want you to have the ability to drop quizzes. So you'll just go through it quickly. If you're on it, you'll find a time to chat with your group mates before then. Google Hangout, fantastic way to do things, right? Who does Google Hangout? Yeah. Who said what's that? What? Okay, Tom, who does not know Google Hangout? Okay, first of all, I'm supposed to be the old person, right? I'm really not. I'm not. But you guys are very disappointing. You can have an online meeting, like via webcam, with like 10 different people in Google Hangouts. You have no excuses to not be working with groups and group projects because you can't read. I don't care if you live in Frederick or what. Get on Google Hangout. Okay, that's all I'm saying. So explore it. It's through Google Plus. If you've got Gmail, you can figure it out. Anyways, up to you. We're making it up next week. Good. So we've been studying cell cycle. we've been studying all the different ways cell cycles regulated with cyclins and CDKs and all those fun things that we talked about. We talked a bit about checkpoints, and then we started talking about S phase as the backdrop for DNA replication. And we started talking about the DNA polymerase structure and function, how it has a proofreading mechanism. And that's where we were at the end of Monday's lecture. I put this picture up on this slide because I really want you guys to get used to looking at this version of the replication fork, because there's a huge disconnect between what is taught and how you understand it and what the current model is. I guess it's not really that current anymore, but this trombone model where it actually the DNA is actually looping out, because you need to remember that this whole, it's not just like polymerases running in separate directions. They're replication forks and they move outwards, and you need to start to think about the polymerase and all the enzymes that are involved in DNA replication working as one large unit. You may have already seen this and it's old news, but last year a lot of the students were like, what? The polymerases don't run in separate directions? So I put that up there because you'll see it now. You'll see it a little later, and then in recitation next week we're going to draw it out in grueling detail, because I really want you guys to think about it closely. Okay, so the different enzymes involved in DNA replication. Tell me if I missed some. I realized I missed one and I had to add it last minute. Polymerase, helicase, primase, ligase, and topoisomerase. I think that's all of them. I forgot topoisomerase. I have to add that one in. So they all kind of work together to do things like open up the DNA, relieve tension, prime the lagging strand, prime the leading strand, and then where you have those Okazaki fragments, kind of stitch them together as you remove the primers. And yeah, topoisomerase to remove tension. Okay, so let's start with talking about polymerase. You know the other thing so I got my review yesterday on my teaching, and one thing that bothered me is they're saying how there's a lot of goofing off in the back row. And I was like, really? I never noticed. And maybe it's because it's so far out there, but I don't know. They saw you, whoever you are, goofing off. Who is playing Nintendo back there? Plays Nintendo. Anyways, okay. So two major classes of polymerases, right? There's processive and distributive, and they serve different roles in DNA synthesis and repair. Processive means they keep going, they don't fall off. That would be something that would be useful for replicating your DNA, right? And then this distributive more has to do with frequent falling off of the polymerase, and those are polymerases that are more used in DNA repair, where you just have to go in and fix a small section. It's very logical, right? Long chromosomes, lots of DNA, processive. You want it to keep going, not to keep falling off, because the efficiency is fairly low if it keeps falling off. So we'll talk about ways that it stays on. And then with repair, you don't need it to stay on for a long time because you're just fixing a few bases, right? Makes sense. Okay, so there you go. I believe this is not in the textbook, so you might, I was looking through it because I'm trying to still merge these two textbooks I've used. And you might have to look this up a little if you don't understand it, but it's a very basic concept, right? So there are different types of polymerases, right? It's not just DNA polymerase. There are replicative polymerases that copy DNA during S phase. So that's the main one we'll be talking about right now. There are these DNA repair polymerases. And this is not just one, they're whole families of polymerases. There are DNA repair polymerases which do exactly that. Bulky adduct bypass / translesion synthesis. That's when you have DNA damage that can't be fixed. There are polymerases that allow you to go past the damage. Bulky adduct means some large molecule got stuck onto the DNA somehow. That's going to obviously interfere with DNA replication. Translesion synthesis, exactly what it sounds like. Lesion is damage, right? Translesion synthesis, go past lesion synthesis, right? You should have no problem understanding what these are just from the names, if you think about it. It bothered me how many people didn't know what DNase was on the exam, because it's in the name. I'm just saying. RNA primer removal and gap repair. Exactly, remember there are these primers that need to be removed and then get filled in. So that's a different polymerase that comes in and does that. And then there's reverse transcriptase, which you see a lot in viruses, but also telomerase. Okay, so lots of types of polymerases. I just want you to appreciate that. It's not one. There's many, and they have many different functions. I'm not going to ask you to memorize polymerases and names of them and classes of them, because that's useless. But I do want you to appreciate the fact that there are many kinds with many functions. There they are. It's a lot, right? If I made you memorize that, would you be happy or sad? Okay, that was a giveaway. Good. I'm glad you'd be sad, because I won't make you do that. Okay, so let's move on from polymerase for a moment. Okay, so we're going to talk about helicases. You guys already know a little bit about helicases, because we talked about it a little with DNA, with chromatin remodeling complexes, because a lot of them had these kind of helicase activity domains, right? So they unwind DNA, right? Why do you need to unwind DNA during DNA replication? Actually, let me get my list out. Michael Berlet.
Oh, can you repeat the question?
Nope. Yes. Why would you need to unwind your DNA? Why would you need a helicase? What's the purpose of unwinding DNA?
Stuff can access it.
So stuff can access it. Okay. I know it's an obvious question. If I don't ask obvious questions, then I don't get the people who don't know the obvious answers. Okay. So why? So does anyone want to add to that? So why would you want to unwind your DNA? What needs to happen? Yeah. Right, so base pairing needs to happen, right? So you need to open your DNA, because part of the whole polymerization reaction requires the appropriate nucleotides added, base pair, okay. Everyone's on the same page, right? Okay. Good, right, so you need access of polymerase to the single strand template for copying. This is just a picture of the HPV helicase. Everyone know what HPV is? Has everyone gotten the vaccine? Do you think men should get that vaccine? Yes. Hopefully one day they'll just give it at birth to everyone. Can you believe HPV encodes its own helicase? Awesome. Okay, so here's helicase again, right? This is another way to look at it. And so what it does is it wraps around. It's a multiple subunit, so multiple proteins that are the same bind together to form this ring complex, and it basically binds. It is loaded around one strand of the melted DNA and kind of moves towards the double-stranded region, opening it up. Let's go a little further into it. So it's hexamer. Hex is how many? 6, right? It's a hexamer of one protein that forms a ring and uses ATP hydrolysis and the associated conformational change. So when you hydrolyze ATP, you're changing the shape of your ATP, right? It's not big, it gets smaller. And it's that actual hydrolysis that causes the movement, the mechanical movement of helicase along the DNA and helps with opening up and melting the double-stranded DNA apart. Yes?
Does a helicase prevent re-hydrogen bonding with the double helix so it doesn't collapse back in on itself?
Well, there are single-strand binding proteins that are involved in making it not. So we're gonna get to that in just a minute. So the question was, does it prevent it from collapsing? Right, because DNA, the double-stranded DNA is extremely stable and it wants to stay together, so you need ATP, ATP and this enzyme to open it up, and then you need proteins that are gonna bind to the single-strand DNA to keep it from closing on itself. Yeah, thank you for bringing that up. Yes?
When you combine DNA in experiment, you have to heat it up. So I'm assuming that there's no heating up in this, but is there damage associated with helicase ripping open your DNA?
Is there damage associated with helicase ripping open your DNA? I guess to think about that, we need to think about exactly what it's doing when it melts. So it's called melting, it's not actually melting, but it is separating the two strands. I think it's just breaking the, it's disrupting the interactions between the bases is all it's doing. I don't think helicase is damaging. I mean, we wouldn't have survived as a species. I don't think so. But good question. Any other questions? Okay. So we're going to stop here and have a little practice problem. The problem's called is protein extrahelicase. So you're studying a protein with unknown function. You've blasted it and found it contains a conserved helicase domain, so you think it must have helicase activity. So you need to design a biochemical assay to test for helicase activity. So I'm giving you a toolbox which contains single-strand DNA, some long fragments and some short fragments that are complementary, kind of like what you see here. See, long and short. ATP, a spectrophotometer, and the knowledge that single-strand DNA and double-stranded DNA absorb light at 260 wavelength at different length, and an agarose gel if you want to evaluate the size of your DNA. So I want you guys to take a few minutes. This is challenging, but not that challenging. Knowing what you know about helicases and what you know about all the stuff I just gave you. Take a few minutes to talk to your friends, and then we will discuss. Yes?
Are we limited to just listening?
No, you can do whatever you want. I give this to point you in a direction, but if you have a better way, go for it. Okay, so talk to your neighbors for a few minutes.
Absorbency, laser beam shooting through here. See if this works. Yeah, you're right. So after put all that
Okay, guys, do we need more time? Or are we already talking about our weekend plan? It's Friday, can you believe it? You guys all can come tomorrow to make up Wednesday. Right? I'll be here. I don't know what I'm doing. Okay. So, I want to know who had an idea based on what we just learned where to go with this? If you did, raise your hand. Okay. Who had no idea where to even start? Okay. Okay. So let's see. So someone tell me where we can start with this. Yes.
I figure what we do is the short and the long complementary strands are going to bind together, wherever. And the helicase is going to open it up. We'll keep it at a temperature where they won't come back together. Then if we run the agarose gel and we have short and long strands, then you're going to know that helicase was actually working apart.
Okay, good. So you can detect. So you can add your mystery protein, and you can detect whether or not you have bound together or separate based on your agarose gel and how many bands come up and what size they are. Okay, who wants to add? There's so many ways to do this. So many ways. That's the thing about science. This guy I worked with at one point would always say there's a million ways to skin a cat, or there's not just one. I don't know. He's always talking about skinning cats. It's so blood curdling, but he's right. There's a lot of ways to do something. Who wants to add to that? I could think of another way. Yes.
Run your initial sample through the spectrophotometer and see if you have mostly double-stranded DNA, and add your protein and link and run it through again and see if you have mostly single-stranded.
Okay, so use your spectrophotometer and analyze your sample, your DNA sample. So I'm guessing you're talking about them bound together, the short and long fragments. So analyze your DNA sample plus minus mystery protein, right? So, and see if there's a difference. See how much single-stranded versus double-stranded DNA you have, because they absorb differently. Yep, that's another way. Yep. Anyone not come up with a novel approach? Those are the people who are going to go into biotech. Who's coming up with a novel approach? Yep. Well, those are the two ways I found. So good. Thank you, guys. Okay. So single-strand DNA binding proteins. They're called SSB. Yeah.
Oh, that's what
Talking about PCR. We don't do that. Never heard of it. Okay, single-strand binding proteins called SSB in bacteria and RPA in eukaryotes. They stabilize the separated DNA strands, which makes a lot of sense because they'll just come right back together because you're not dealing with this super high temperature or anything that's going to keep them apart. They're just going to want to come right back together. Yeah. So topoisomerase removes the supercoils from DNA created by unwinding. So you can imagine, and I forgot to bring it, I did a thing last year where I had some yarn. No yarn. Sometimes you can get multiple threads that wind around each other, but sometimes you can just get one with two. So I have one with two. I'll try to remember to bring it on Monday. Where if you pull it, it's going to get super coiled, right? You can imagine you pull the two pieces of yarn apart. It's going to super coil, and that's not good because it introduces torsional stress. Yes?
A question with that. Why would it have to be a model of pulling apart rather than helicase rotating around the DNA to eliminate tensional stress?
So the question is, why is it a model of pulling apart versus the helicase rotating around the DNA to, well, so the thing that you have to think about is that you're separating something that's wound around each other. So if you just turn around, I can see, still though, the other end, so the other thing to think about is the other end is kind of fixed in a way because it's part of this large amount of DNA. And so you would need the other end to flip, or something would need to be happening. If you've got a fixed point and something tightly wound, opening, even if you wind your, I don't know. I can't answer that question in a way that satisfies me. But it doesn't
Right, it doesn't.
do that.
As long as you stabilize the lagging pieces with your proteins, so there wouldn't be recoil in the back, that would cause your heel case to tighten up as it moves along in that model.
Okay, well, you know what? Let's work on that. We'll publish a paper together. I have no idea why. But feel free to come and discuss, because it's an interesting discussion. Yes?
Is it like that for like both, like for bacteria and eukaryotes? Because if that's maybe not, then maybe it's just like for eukaryotes, then maybe it could be like because eukaryotes have multiple artificial organs, then it'd be more prone to mistake if the little case goes around. But they'll have to go around it for all of the replication organs going either direction.
So you asking if topoisomerase is in prokaryotic and eukaryotic cells? Kind of? Huh? Something like that? Yeah. So you're thinking about multiple origins in eukaryotic cells. You're going to have more of a mess versus in bacteria. Yeah, I'm not sure how to answer the question. You do end up with a mess on both of them. We'll see in a minute. And there's different topoisomerases to deal with the different types of messes. Yes.
Maybe it's just that because the glob's on there, it doesn't have any, like these things can't think for themselves, so it's just based on interactions with the DNA strand, so it's just stuck on there, goes out, flips it up.
Yep. So maybe the enzyme just binds and moves forward, and that's it. And it's not like it's thinking for itself. Yeah, I'll go with that too. So we need to deal. There's supercoiling that happens from the activity of helicase, whatever reason it does. That's just how it is, and there needs to be a way to deal with it. So there's topoisomerase to deal with that, and there's two kinds. There's the class 1, class 2. So topoisomerase 1b here, it actually, what it does is, so here's your double helix. Doesn't look like a helix at all here. And then your enzyme, your topoisomerase comes along, and it has a tyrosine in its active site, and it binds to the phosphate backbone and creates a bond there and breaks the bond here. And so it makes a single strand cut. And then the torsional stress powers the backwards rotating. So it actually rotates around the single strand that remains bound. And then it closes it back up. And so that's one way of relieving torsional stress. Yes?
Does that say flicky?
Huh?
The last word on the top, very, very top.
Nicking.
Nicking. Okay.
Nicking. Yeah, the rabbit ears are in the way. Sorry about that. Yeah, topoisomerase 1b rotates the DNA by nicking one strand through a transient phosphodiester bond between the enzyme and DNA. What? Okay, I'm so glad the mind has been blown in the front row. We're going to try to blow minds on the left and the right and then the back. Do you think we can do that? We can. Okay. So, rotating, very cool. So the torsional stress is already there, so it doesn't require ATP because there's all this stress and it just flips back the other way. You can imagine if you've got this thread pulled tight and there's all this stress and you nick one, it's just going to unravel, right? Okay, so then there's these type 2 topoisomerases, and they work differently. They relieve torsional stress by introducing a double-stranded break. So if you're thinking to yourself, what's the difference? One's a single-stranded break, one's a double-stranded break. One does not require ATP, one requires ATP. So what this does, and I've got a few ways of showing this, because this figure always confuses people. But this is 2 strands of DNA, it's not single-stranded, it's actually double-stranded DNA. Because remember, it's a big mess, right? They're all up next to each other, and all this replication is going on. So sometimes you'll have them stuck on either side of each other. And so what it does is it binds one side and passes and makes a double strand break and passes. So it's binding to the pink and cutting it and passing the blue through. So, and so ATP hydrolysis is what induces the conformational change required for the passing through, the binding and then the passing through. Type 1A enzymes also use this. It's called strand passage, but it's the passage of a strand through a break. It's a passage of a strand through a break in a single strand. But don't worry about that detail. So here's another way to look at it. So you can have supercoiled DNA. So this is looking at it as a bacterial chromosome, and this is where a lot of this work was originally done. So you can end up with supercoiled. This is not really very supercoiled. It's just like one twist. But in true reality, it would be really supercoiled. And then you could also have this catenated. Remember when you're replicating circular DNA, you kind of end up with them intertwined within each other. So they're linked through like chains, like a chain link. And so you need to resolve both of those. And so you can either relax supercoils or decatenate, which is where you resolve the kind of linking of the genome using these type 2 topoisomerases. And so this is your enzyme, and it will bind to one. They're calling it a G segment. Don't worry about genes. You just realize there's 2 strands that need to be passed. One needs to be passed through the other. This is from a Nature review, so they always like to complicate things by giving different names to things. But so it binds to one, and then it captures this other strand of DNA with part of the enzyme, and then it cuts it and passes it through. See, it's up here, and then it passes it through the blue strand and releases it on the other side. And the ATP binds and is hydrolyzed. So here it's open and it binds, and that's what results in it being closed. It binds and then it's hydrolyzed, and then it releases the ADP, and then it's open again. So that different binding states of the ATP result in the movement of the enzyme. Okay, so it's really just about passing one strand past another by making a break. And then the mechanism is similar because it's also creating a transient bond between a tyrosine and the phosphate backbone. Yes?
So the blue strand isn't cut or broken, it's just the red one?
The blue strand is the one being cut, the red one's being passed through. So here, if you look right here, see here, it's being bound. And then here, it actually, there's a transient bond between the enzyme, the subunit of the enzyme on this side, and the subunit of the enzyme on this side, and the phosphate backbone of the blue. And so that's what's doing the double strand break. And then it's passing the red one through. Okay? Any questions about this? Okay. So that's topoisomerase. We talked about helicase. We talked about topoisomerase. And so I told you that replicative polymerases are highly processive, meaning they stay on the DNA for long periods of time, which they need to do because there's a lot of DNA to be replicating, right? And the reason that they're highly processive has to do with the sliding claim. So only the DNA polymerase that replicates the chromosomes interacts with this sliding claim, and that's why it's so highly processive. And so this is what the sliding claim looks like. It's a ring. A bacterial one has 2 subunits, and the eukaryotic one has 3 subunits. It's also 6 subunits altogether, 3 dimers or 2 trimers. What else can I tell you? This is how it looks when it's bound to the DNA. So here, the white is the DNA, and you can see the sliding clamp is completely wrapped around the DNA. So you got to think, how do we get on there, right? And that's due to the activity of the clamp loader, that it binds tightly. Now this I thought was very interesting when I learned this. This is the coolest class for me to teach because I kind of get to learn everything over again every year. The clamp loader binds tightly to the primer template junction. Now, so if you remember what you've learned about DNA replication before, you need a primer to start your DNA replication, right? Because you need that OH. So it makes sense that your clamp loader is going to bind most tightly to your primer template junction, so where your primer is binding to your template, because that's where you need to load your sliding clamp on and recruit your polymerase and do your DNA replication, right? I thought that was cool. Yeah, so there's a clamp loader, loads on the clamp through this mechanism. So it also uses ATP because, again, you have movement. Whenever you have a protein moving, you always need to think about ATP hydrolysis and how the size of the ATP is what is responsible for the movement of the protein. So here's your sliding clamp, and your clamp loader, and your ATP, your DNA, and your clamp loader. Actually, so you've got your sliding clamp and it's open. Your clamp loader puts it onto the DNA and closes it, and then it leaves, and then DNA polymerase is recruited. Okay? Questions about this? Comments? Concerns? Anyone want to talk about what they're doing this weekend? I heard someone say studying. That's good. That's all you should do. My PhD advisor once ran into one of the girls in my lab out at brunch. Like, they went out for dim sum over the weekend, and they both saw each other. And she didn't get good data the following week. And he was like, if you weren't so busy going out to eat, you probably would have gotten good data. Like, research is so important that eating is secondary. That was always fun. Okay. Clamp loader is released, right, once the clamp is assembled, but it stays nearby. Okay, it's not just gone and that's that. 'Cause you have to think about, you've got your leading strand and your lagging strand, and your leading strand is going to be highly processive, but your lagging strand is going to keep having to release and come back on, and release and come back on, because remember, it's going to jump to and do each Okazaki fragment separately. So your clamp loader is actually hanging out there waiting to put the clamp back on. Okay, are we having fun? Yeah, okay, I see one person nodding, as long as one person's having fun. Okay, so the clamp loader associates with a clamp after ATP binding. It opens the clamp, ATP hydrolysis closes the clamp and releases the clamp loader, right? So here's your clamp loader, and then here's your clamp, and you've got it binding, and then it's released. Okay, and then the clamp recruits DNA polymerase. Okay, DNA replication mechanism. Very basic, right? Initiation, priming, leading, lagging strand synthesis, replication, fork, termination. Done. That's it. You just need to understand it in a little more detail than that, but those are the main parts. So when you're starting to think about studying, not that you really want to study again, but you're going to have to, these are the main categories. So initiation needs to happen. Now, we kind of talked about it from a cell cycle regulation perspective, right? You've got this origin recognition complex that binds to the origin, right? Remember that? This was about a week ago, right? It's loaded long before DNA replication is ready to begin, right? What phase of the cell cycle does the origin recognition complex bind to the origin? What part of the cell cycle is that? Daniel Iorinde. Before S-phase, there you go, right? G1, even late mitosis, yeah. Complex is activated, so you've got this whole complex sitting there for a long time. It's activated. Do we remember how it's activated? Through the activity of what? Amanda Talbot? Amanda? Amanda. Okay, I feel like I've called on her before. Okay. Charles Hawkins, Chip? I don't know. So the origin recognition complex binds to the origin and it's hanging out, staying around. How is it activated? Through the activity of what? That we talked about already. CDK, right? A CDK, right? SCDKs, right? Because S cyclins came up and activated the SCDKs. Trying to bring this all together, okay? And something that, another way to bring in stuff we've talked about before is that the ORC is recruited to origins with hyperacetylated histone tails. Interesting, huh? Why is that interesting? Why would the origin have hyperacetylated histone tails? Oh my God, am I making you guys think about something you already had us on? Did you do a brain dump? No brain dumping left. Yun Lee. Did you just take a bite of something? Should I go to someone else because you have a full mouth? Okay.
So,
So I said, fun fact of the day. The origin recognition, ORC, origin recognition complex is recruited to origins with hyperacetylated histone tails.
so we have more open DNA.
More open DNA. Is it logical you would want your DNA open prior to DNA replication? Do you think it needs to be in heterochromatin? Highly condensed? No, that's crazy. Right, exactly. I don't know. Every now and then I want you to try to join all these things we learned together. Every now and then. Okay. So we're going to end with this thought. DNA polymerase functions as part of a large multi-protein complex. Okay, it's not, huh? Nope. Let's get to this next figure, and you'll see what I mean. So this is what I showed you earlier on, okay? It's this huge protein complex, right? So let me just orient you to what you're looking at, because it looks like a big mess, and it kind of is a big mess, right? So you've got your parental DNA helix, you've got your helicase that's running this way, right, and opening up that helix, and then you've got interacting with all of this, you've got your sliding clamp and clamp loader, which is hanging out because remember, it's going to have to keep coming off and going back on over here because this is your lagging strand, this is your leading strand, so it's fairly simple. As your helix opens up, your polymerase and your clamp are hanging out and just moving this way, moving to your right. This way is a little bit more complicated. And I want you to really start thinking about this because, like I said, when we started the lecture, people think of DNA polymerase as just going in separate directions, which it does not. It's always involved with this complex. With your helicase, your clamp loader, your polymerase, those are always kind of in a group together, working as a holoenzyme. So one large complex of proteins functioning as an enzyme. Okay? It's like a machine. And so here's your leading strand, and then here's your lagging strand. And this gets complicated, right? Because you've got your single-stranded DNA coming here and looping back around this way, and going that way. And so that's how it works, because this is going to fill in up to here, and then it's going to release, and then this is going to move out, this is going to move around, those single-strand binding proteins are going to come off. It's going to bind to the next primer, which is up here, and it's going to fill this in. And so that's how it all stays together. And this is really, if people leave recitation when we do this with headaches, because it's really, maybe it's simple for you, but I think it's very complicated to look at. Who thinks it's simple? Great, you will lead recitation next week, because I'm going to get confused. I get confused every time I look at it, because it's a lot to look at. But it's just trying to understand exactly what is moving in what direction. Because you want to remember that this whole complex is moving to the right. It's opening up, going through this way, right? But you have the leading and lagging strand where the polymerase is kind of moving in opposite direction, but still staying together. Major concept to kind of get past. This is the trombone model. Does it look like a trombone? I think it's this thing. Yeah, I don't know. I was talking to Dr. Master. She's like, I can't believe you're making them learn that. It's complicated, but you guys are gonna love it. Okay, so go away, and I'll see
Are we gonna read?
at the end of the day.
Oh, online.
Yeah.
Yeah.